Hi, everybody. Welcome so much for joining us for today's Live Longer, Live Stronger lecture. My name is Andrea Decker, and I have the pleasure of representing the Fleet Science Center and the Sanford Brennan Previs Medical Discovery Institute. These Live Longer, Live Stronger is a 5-part lecture series in partnership with the Sanford Brennan Previs Medical Discovery Institute, and we're very excited that they have been such a wonderful partner for us for many years now, and we're glad that we can continue this partnership with this amazing talk series. So today we're going to look at Alzheimer's risk factors with Dr. Timothy Huang, and just a couple of things at the beginning of the talk, just some household items, I guess. We're going to go through the presentation and then we'll have a Q&A afterwards. The way that you can engage with our Q&A is through the Q&A function in this Zoom webinar. So when you have questions, just type it in. Whenever it pops into your mind, you don't have to wait until the Q&A. And this way we get to it when we are at the point of the Q&A. And also during the Q&A, I will start a poll. We always like to survey our audience to make sure that our event is good and that we improve on the event. We also like to learn more about our audience. So if you could take a second to fill that out while you listen to the Q&A and ask your questions, that would be wonderful. All right, I don't want to take too much time away from our talk, so I'm going to turn it over to Dr. Huang now.
Thank you very much, Andrea. So let me share my screen here. All right, so we're going to talk a little bit about the Alzheimer's disease symptoms, and we're going to talk about the impact of AD, Alzheimer's disease. I might call Alzheimer's AD sometimes. We're also going to talk about how Alzheimer's relates to other dementia disorders and how do we distinguish Alzheimer's from other dementias. Secondly, we're going to talk about the 2 different forms of Alzheimer's disease. So this would be the inherited familial forms, which involves genes such as APP, and the more common form of Alzheimer's, which is the sporadic form that involves genes such as TREM2 and ApoE. And lastly, we'll talk about how we approach Alzheimer's disease in the lab. That is to say, how do we find if something is protective or aggravates the Alzheimer's state? And basically, we use mice and cellular models to study different pathways in Alzheimer's disease. So first of all, let's talk about what is Alzheimer's disease. So the National Institutes of Health describes Alzheimer's disease as an irreversible, progressive brain disorder that slowly destroys memory. So basically, it impedes thinking. And eventually, things that you do on a day-to-day basis, they become very difficult or impossible. So in most people with Alzheimer's disease, the onset of age of onset is about mid-60s, around 65. And Alzheimer's is the most common cause of dementia in adults. So dementia is a bunch of disorders that result from either injury or damaging factors that impairs brain function. So Alzheimer's is one of these dementia disorders that takes the biggest piece of the pie. So up to about 80% of dementias are end up being diagnosed as Alzheimer's. Whereas there's other dementia forms, for example, vascular dementia or dementia with Lewy bodies, frontotemporal dementia. These are all smaller proportions compared to dementia patients, which are mostly Alzheimer's disease. So Alzheimer's is projected to be a serious problem sociologically as well. And the numbers that are projected between now and 30 years from now are quite staggering. So it is estimated perhaps around 500 million of Americans are currently living with Alzheimer's disease. And this is projected to increase to 14 million in 30 years. And so the cost of care is also quite staggering. So the current cost to society is about 305 billion, and this is projected to increase to 1.1 trillion. And surprisingly, it seems to be estimated that 1 in 3 seniors will die from Alzheimer's or some type of form of dementia. And most importantly, currently there's no drugs that are currently available that will reverse or cure Alzheimer's. There's only drugs that are prescribed to basically treat the symptoms of Alzheimer's. So as mentioned previously, so most individuals will show symptoms at around age 65. So some of these symptoms include, the biggest one is, of course, memory loss. So people will have an inability to remember recent events or recall new information. Individuals with Alzheimer's will also have difficulty with doing things they've been doing their entire lives, so completing routine tasks. They will also have difficulty with telling where they are, what day it is, what month it is, also what time it is. They might also have difficulty with speech or writing. And they will also have potentially personality changes. So they might become socially withdrawn, they might have mood changes. So these are all manifestations of Alzheimer's or Alzheimer's-like dementia. So Alzheimer's has become more of a recent problem with the aging population, and this is more something that has come up more recently. So in the 1800s, the life expectancy was quite low. So just take this with a little bit of a grain of salt, because the consensus methods during this time is probably not great, and there's a high rate of deaths at birth. So even within the last half century, the life expectancy seems to be about 50 years of age. So now we're seeing an aging population where people are expected to live past 70. So this is kind of alarming to us, because the age of onset for the sporadic form of Alzheimer's is past age 65. So here we are. So Alzheimer's is going to be a huge impending problem. It's devastating to the patient itself. It's also devastating to their families, and basically with the aging population that's going to bloom, it's going to have a huge impact on society. So it's important for us to understand the disease itself, and how do we distinguish Alzheimer's dementia from other disorders, dementia-like disorders. So decline in mental function has been documented in the early ages, for example, in Greek and Roman history, the medieval era. And basically mental decline was thought to be just a general function of aging, a consequence of age. So it was not until the 1800s that physicians such as Philippe Pinel, a French did I say physicist? Physician, I'm sorry, who characterized the dementia more carefully. And basically, he was a huge advocate for the humane treatment of dementia patients. And during this time, pathologists or physicians, they have also noticed that with dementia is associated with the wasting away of the brain, or brain atrophy. So healthy tissue seems to remain intact, whereas during dementia, the tissue appears to degrade. So in 1906, Dr. Alzheimer was caring for one of his patients, August Dieter, in Germany. And this was a woman in her 50s who showed dementia-like symptoms. When August passed away, Dr. Alzheimer performed autopsy on her brain, and he saw what we now know are hallmarks of Alzheimer's disease. And these are formations within the brain that he described as plaques and tangles. So what are plaques and tangles? So plaques are these clumps of entities within a diseased brain. So in the 80s, George Glenner over at UCSD and an Australian, Colin Masters, they isolated the component within these plaques. And what they identified was a short 40 to 42 amino acid peptide that's called A beta or beta amyloid. So this is the primary constituent of these plaques that we can just call these beta amyloid or A beta plaques. So later on, Several other groups have identified the primary component of these tangles, these twisted structures within the diseased brain, and they identified tau as the modified form of tau as the primary component of these tangle structures. So plaques and tangles, they define different classes of neurodegenerative disorders. So there's just Alzheimer's disease, which has both plaques and tangles. But Alzheimer's falls under a more generalized class of diseases called tauopathies. And these are all diseases that have this tau pathology or these tau tangles within the brain. There are dementias that only have these tangles. So these are disorders such as cortical basal dementia, frontotemporal dementia, or progressive supranuclear palsy. So these are all disorders that only have tau, and there's no beta amyloid, these plaques. So what we can notice here is that basically the frequency of these tau-only disorders are only a fraction of Alzheimer's. So this is where we're at in terms of, we know that beta amyloid plaques are specific to Alzheimer's disease. So basically they define Alzheimer's. So Alzheimer's is the most prominent neurodegenerative dementia disorder. So what do plaques have to do with Alzheimer's? Do they have something to do with the degradation of the brain function? So we have an idea that it's actually the protein, so the beta amyloid within the plaques that might have some toxic effects to the brain to trigger the neurodegenerative process. And so we're going to talk a little bit about the genes that are associated with Alzheimer's disease, so the inherited form and the non-inherited sporadic forms of Alzheimer's and how they relate to beta amyloid toxicity. So this is an ongoing story as various research groups come to try to understand how beta amyloid is related to neurodegeneration. And so we need to discuss about the two primary forms of Alzheimer's disease. There's an inherited form, which involves the gene APP, and a sporadic form, which involves other genes. So the inherited form of Alzheimer's disease results in early onset. So individuals will start to see symptoms at about age 40. And each carrier of a mutant of, for example, the gene APP will almost invariably show symptoms at age 40. So this is what we say is very penetrant. These dominant inherited forms almost always manifest in Alzheimer's symptoms at an early age. And what I'll discuss later is this is directly related to the production of beta amyloid. However, these cases are very rare. So most of the cases in Alzheimer's disease are sporadic and they're late onset. So individuals begin to manifest symptoms at about age 65 or later. So there are gene variants that have been identified in genes such as TREM2 or ApoE that can increase the odds of risk of Alzheimer's onset after age 65. But the relationship to A beta is a little bit more complicated. So basically, the relationship might be something to have to do to how the brain responds to beta amyloid rather than the direct production of beta amyloid itself. So we're going to need to talk about the inherited familial gene APP. So this is the amyloid precursor protein. So in 1987, basically, they cloned the full-length gene of which represents a part of which basically the beta amyloid portion is embedded within a region of this full-length gene. And within the 90s, they characterized enzymes that cut this full-length APP protein that's made from the APP gene into this 40 amino acid or 40 to 42 amino acid fragment. So in the 90s and onwards, they identified gene variants that accelerate or enhance the production of this small beta amyloid peptide. And these are familial inherited early onset forms of Alzheimer's disease. So these mutations will almost invariably lead to early onset of Alzheimer's. So this is very strong evidence that beta amyloid is doing something that's hopefully not good for the brain. So the question is beta amyloid toxic? So neurons, they communicate through each other through synapses. So neurons form an interconnected network, and they basically communicate through a presynaptic terminal, which then throws out neurotransmitters, which is received by a receiving neuron at a postsynaptic terminal. And so what we know is that beta amyloid is sticky, especially if beta amyloid is in clusters. And it tends to interfere with this communication process between different neurons that are connected in a network. We also know that in mice, if we put in a gene, a human gene with one of these mutational variants, these inherited mutational variants for APP, we can also produce more beta amyloid. And the effect is basically the mice have problems with memory in addition to the formation of these beta amyloid plaques. So this is good evidence that there's something to do with this buildup of beta amyloid that is having toxic effects in the brain. So there has been significant effort within the last 2 or 3 decades to identify strategies in order to decrease the amount of beta amyloid load in the brain. So these include use of inhibitors against these enzymes that cut the full-length APP protein into this beta amyloid fragment. So many of these inhibitors have gone to clinical trial, and so far all of them have failed. There's also been strategies, so some antibody based strategy which target beta amyloid to enhance their clearance or to decrease their levels in the brain. And so far, these have also largely failed in clinical trials. There hasn't been really a single success story in terms of the clinical trials with the AD-associated drugs. That's not to say that we haven't given up. According to the NIH, basically, the NIH is supporting 272 dementia drug trials in the clinic, and about 80 to 100 of these are targeting specifically Alzheimer's disease. So we'll see if any one of these might have success down the road. But we need to ask the question, well, why is it with all these extensive efforts that we haven't seen really any success in the clinic, that reducing beta amyloid doesn't really seem to have effects? Well, these things that are anticipated to reduce beta amyloid in the brain, they don't seem to have any effect. So one possibility is we're treating these patients too late. So timing is everything. You can just imagine that we can prevent a fire by pouring a bucket of water on a match, but if we pour a bucket of water into a raging forest fire, that's not going to do too much. So we have a similar problem with Alzheimer's disease. So the levels of beta amyloid that build up in the brain occur basically 1 or 2 decades before individuals see any symptoms. So basically there's no memory problems for 1 or 2 decades. And that's associated with a significant buildup of beta amyloid. So when the beta amyloid almost tops out, that's basically when we're starting to see mild to moderate symptoms. And this is basically where we're treating the patient. So it might be a little bit too late. So one way to get around this, so new clinical trials are targeting these dominant inherited forms of Alzheimer's. So we know that these patients will accumulate beta amyloid. There's almost an invariable chance that these patients will have early onset AD. So hopefully with in using these patients rather than sporadic patients, we might be able to have a better idea of what drugs might be able to have a better effect on Alzheimer's symptoms. So we'll talk a little bit about the sporadic form of Alzheimer's disease. So this is a little bit more tricky. Basically, when the human genome was sequenced in about year 2000, we come to understand that individuals between myself and yourselves are identical at the nucleotide level, about 99.9%. So there's a 0.1% variation between different nucleotides between individuals. We call these variations polymorphisms. So for example, I might have a T1 at gene X or nearby gene X, whereas another individual might have a C. So that's enables us to ask the question whether if T carriers are more susceptible to, if there are more T carriers that have Alzheimer's disease compared to individuals that don't have Alzheimer's disease. So this allows us to identify single gene factors that increase the risk of Alzheimer's onset. And we've been doing this for about a decade now, scanning the genome for variations in genes that might increase the risk of Alzheimer's onset. And it's surprising that the genes that are identified with variations that are associated with greater risk are enriched in immune cell types in the brain. So these are microglia primarily and astrocytes. So basically genes such as CD3, 32, TREM2, and ApoE can increase the chances of AD risk after age 65. So I just want to go into microglia and neurons and how they integrate with each other into the function of the brain. So basically, we can imagine that the brain is an interconnected network of neurons, not unlike the internet. So basically, the internet is an interconnected node of different computer terminals that allow us to communicate with each other. So the brain also needs to communicate to different parts of the body and within itself to store memories. And neurons, basically, they can talk to each other, and that's how we function. So basically, we taste because neurons talk to each other, and we record memories and listen to music. So, like the internet, the neurons need some type of support system to fix damage or sense damaging agents and mediate repair. And so basically something like an IT support system in order to sense and repair damage. So these are microglia. So these are innate immune cells within the brain that sense damaging agents. They will clear away debris or damaged cells, and they will have support function for the neurons. And it's really fascinating. There are other cell types that actually also interconnectedly mediate dysfunction, including oligodendrocytes and astrocytes, which we won't go into today, but microglia have an essential function in also surveying the brain environment for substances that might be toxic, for example, beta amyloid. And so microglia, they turn off and on in response to damage or disease. And so basically when they're turning off and on, they signify their on or off states through the activation of various genes. So I'm going to borrow a clever analogy from Chris Glass over at UCSD, is that we can tell what the microglia are actually thinking by the genes that express. In other words, for example, if a microglia is expressing a gene, making an mRNA that's about to make a protein that is involved in cell motility, we can think that the microglia are thinking they want to move. When this mRNA is translated into protein, this is like the microglia saying that it's about to mediate some function, which is like the microglia saying that they're about to move. And this particular protein, when it binds to ATP or ADP in an extracellular environment, it activates and this enacts its motility. So this can trigger a behavior, and we can measure that in the cell. So basically, this is a movie from Bing Zhu in the lab. He shows that differentiated microglia from embryonic cells can move towards the ATP source in a pipette tube. So microglia, they appear very differently. Their shape when they're at rest or when they're activated. And they have a very different language when they're in the resting state and also when they're in the disease state. So these are fluorescence images from the lab showing that microglia, they have these extended filaments that kind of survey the brain environment for damaging agents. And when Yinjin added A-beta to mouse brain, basically these microglia become activated. Their shape becomes very condensed. Their cell body expands. So they not only have a different shape, but they also have a different language. So they make a different subset of mRNAs associated in response to some type of damaging agent. So we can imagine that the normal microglia response to damage is that they sense some type of toxic component. This enacts microglia activation, and the microglia will mount a response. So basically, they will secrete cytokines that will recruit other microglia to help repair the environment, to take up the A beta and degrade it, and to basically clear the toxic environment. And this will allow the microglia to return to a resting state. So the Alzheimer's gene variants, interestingly, they can affect how the microglia think. So basically, the different mRNA transcripts that are associated with the disease state, they're affected by the expression of these AD risk genes themselves. So when microglia become activated, they actually turn on genes such as TREM2 or ApoE, which are AD risk genes. And this will affect the expression of other genes. So it will affect how the microglia is thinking. And at the same time, these Alzheimer's gene variants, they will also affect how the microglia behaves within the disease environment. So these are beautiful images from Jaime Grenzendler's lab. He shows that basically in a TREM2 mutant, the microglia does not associate it as well with beta amyloid plaques as an AD patient, which does not have a TREM2 mutation. So this is suggesting that neuroinflammation is a very integral part of the Alzheimer's process, neurodegeneration associated with Alzheimer's. This is very apparent that once A beta levels build up, we start to see significant activation of microglia and astrocytes. So what we might envision that's happening is basically the brain is exposed to, and the microglia are exposed to beta amyloid, and the excessive beta amyloid buildup results in the microglia activation, which can also activate other cell types and also activate, neighboring microglia cells, which then starts a process of taking apart neuronal synaptic networks, for example, and this goes on to a point where the damage becomes irreversible. So basically, the communication network between neurons start to break down, and then that's when memory deficits are thought to start to manifest. So basically, we need to try to find some type of strategies to try to slow or reverse Alzheimer's onset. So I just want to introduce some strategies we're taking in the lab using mouse models and using differentiated embryonic stem cells to try to find out how Alzheimer's disease risk variants can alter the behavior of different cells and our response to beta amyloid in mouse models. So one of the Alzheimer's disease risk genes that we look at is SORLA. So SORLA has various mutants that have been identified that are enriched in AD. So basically, this is saying that there's probably some defects in SORLA that are manifesting in mental memory impairment, for example. So one strategy is that that we might want to take is that if we increase the dosage of the sorla gene, this might have some protective effects. So basically, the brain might be able to cope with more beta amyloid, or it might reduce the beta amyloid levels itself. And the way we go around testing this is we use the mouse model. So this assay here is called the Morris water maze. So we can test how well mouse is able to remember something. So the mouse does not want to drown. So basically we give it a platform it can't see, and we force it to use these markings on a pool wall to remember where this platform is. And we can ask the question, if there's more beta amyloid within the mouse brain, how well is the mouse able to remember where we put the platform? And so we find that when we inject beta amyloid in the form of oligomers, or small clumps, into the mouse brain, the mouse isn't able to remember where we put the platform. But if we take a mouse that has an extra human gene copy of the sorla gene, we find that these mice are able to find the platform. So they're able to remember better despite the beta amyloid that we inject into the mouse brain. So I just have one more short story that we have been working on in the lab. So this is from our microglia group. So they've been studying how these Alzheimer's disease risk gene variants affect microglia thought and behavior. And so they are able to integrate Alzheimer's gene mutations into embryonic stem cells and differentiate these embryonic stem cells into human microglia-like cells. And then they're able to ask the question, how does this affect the expression of other genes? So what are the mRNA populations that are being made within these microglia at a global level? So in other words, how does this mutant gene affect how microglia think? So at the same time, they can take these microglia and ask the question, well, how does microglia interact, behave in a disease environment? So they can implant these microglia that have these gene mutations into a mouse that have a gene expressing that's making this beta amyloid protein, and they can then look at how these microglia are able to interact with the beta amyloid and how these microglia are able to clear away the beta amyloid in the brain. So in essence, basically what we're doing is we're trying to translate what the microglia are thinking, so basically the expression patterns of the mRNA in the microglia, and correlate that with what the microglia are actually doing within the brain. So we can identify protective activities in the microglia. So basically taking up the A-beta, the beta amyloid, clearing it away, associate with A-beta plaques. And we can, if we find a correlation between these two, we can try to do an experiment we call repositioning. So basically we can find drugs that make the microglia think they're in a state that they actually do something protective within the Alzheimer's brain. So that's kind of the endpoint that we're trying to reach within these studies. So basically, where do we go from here? Well, I mean, what should we do now in order to try to prevent dementia or the buildup of beta amyloid? So it's just basically common sense that if you can take care of yourself, the better you take care of yourself, the better your brain will take care of you. So leading a very healthy lifestyle, that will be important to reduce your chances of Alzheimer's. And there's protective activities that have been shown that are extremely beneficial, especially exercise. So exercise is one of the most protective activities that you can do to try to protect yourself from sporadic Alzheimer's. Of course, it's important to look after your heart. Your brain is very sensitive to fluctuations in oxygen levels, so it's very important for you to take care of your circulatory system and to follow a healthy diet as well, and to enjoy social activity, of course. And so I'm sure there are members of the audience that are also taking care of older relative So it's estimated that maybe about 15 million Americans are providing unpaid care. And so that's a lot of stress. So you guys also have to take care of yourselves. So basically, you need to ask for help when you need it. There's support groups that you can join, and just try to take a break whenever you can. So there's resources out there that can help. I found that the Alzheimer's Association website is very helpful. They also have a hotline you can call if you have any questions. So I just want to end by thanking all of you for attending. This was a really great experience for me. I'd like to also acknowledge current members of the lab and previous members. So this is basically us a couple of years ago. This is us right before the pandemic, and this is us now. So we have numerous lab members that have left for other opportunities during the pandemic. And, we're always in need of a helping hand, so we're currently trying to recruit new lab members. I also want to thank various collaborators throughout the projects within the lab at SBP, also UCSD, UC Irvine, Harvard, and Max Delbruck, and also acknowledge our funding sources. So NIH, Cure Alzheimer's, and Alzheimer's San Diego. Okay, so this is my email. If you have any questions, please feel free to email me. This is our lab website. Please feel free to check it out.
Wonderful. Thank you so much, Timothy. Such amazing work, and we already have some great questions. So if you wouldn't mind stop sharing your screen, Timothy.
Yeah. Okay.
Thank you so much. That just makes it easier for people to see you when we get to the Q&A, which I'm going to kick off right now. So the first question we have is there any positive use in the body of beta amyloids, or is it only toxic?
Yeah, so I've been asked that question before, and there is some evidence that beta amyloid might be antimicrobial. So having said that, this is an answer that I've given previously. I have a feeling that if we don't have any beta amyloid in our bodies, that's not going to have any effect. Part of the reason is because the gene knockouts in mice, they have no effect basically on the mice. The mice are completely normal. Having said that, so the beta amyloid in mice and human are very different. So that still remains up in the air. So I'm just basically trying to speculate based on current research. So there has been some evidence that beta amyloid has some antimicrobial properties.
Thank you for that. Can we skip back a little bit and explain the term peptide for a second?
Yeah, peptide is just a small piece of protein. So beta amyloid is just a 40 amino acid block from the amyloid precursor protein, which is about 800 or 900 amino acids. So it gets cut into these small pieces.
Thank you for that. I know that just helps a little bit with the background. And another question is, do you think there's any hope of progress in a chemical diagnostic test that measures amount of beta amyloids in the blood or the brain? I think it's interesting as we get more and more to personalized medicine. I'm wondering if that just becomes a standard test with your physician.
Right. So the problem with beta amyloid is it starts building up very early. So I'm not quite sure if you guys are aware of this. So there's a compound called Pittsburgh B that we can actually visualize how much beta amyloid is building up with the brain. So I think another test that the clinicians do is they take cerebral spinal fluids, do a spinal tap, and basically they can measure components that might give us a clue into the biomarkers that might be indicating that someone might have Alzheimer's. So in my mind, they're working on that. So there are components such as neurofilament that do go up in patients that eventually end up with Alzheimer's disease. But in terms of something really definitive, that's kind of up in the air right now.
So let's say they come up with a test and we get there. It's a regular test, once you get your physical, and that's part of the physical. But what does that mean in terms of treatment? You said there's no cure or anything available yet. So even if we know, how could that help us?
Right. So I'm just waiting for, as I said, so basically they're starting drug trials where they're taking the inherited forms. So they have identified and recruited families with Alzheimer's. So they know someone has a gene and they know they're going to develop beta amyloid. And basically, if they can fix that problem, then with some type of drug, then basically we know the treatment regimen. So my feeling is that comes first. We need a success of some type of treatment before we can say, okay, if we have a good diagnostic, then we can go and treat the patient. So I think this happens sequentially. But it'd be nice if we could have a diagnostic anyways, even if we don't have a treatment. So I think that would be absolutely, very useful.
Thank you. The next question is, have you heard of gamma light and 40 megahertz sound gaining credence as an Alzheimer's therapy? Is that something you're aware of?
Yeah, so this is some work from Li Tai's lab over at MIT. So she's done some really fantastic work showing that light flashes at a certain gamma frequency can have some protective effects in the brain. This is partially from modulating the microglia response. So I'm not sure what really came out from that, but I do know that they've also are starting to develop other types of stimuli, for example, auditory gamma frequency modulation and trying to modulate this Alzheimer's onset. So I think that there's hope. I think it's really cool. When I read that, it was like, it totally blew my mind. So here's hoping that that might turn out to be something so non-invasive. I mean, that would be really cool.
Yeah, that would be great. And we have two questions actually that ask sort of the same thing. Is there evidence of environmental factors causing Alzheimer's, such as chemicals in water, food, or air pollution, things like that?
I'm sorry that I have to think about this for a second. I'm kind of,
That's
running
okay.
through all the articles that I've read in my mind that, right now I can't really think of something that's completely definitive that, for example, if you um, are exposed to some type of carcinogen that, this induces Alzheimer's. I'm sure something will come up in my mind later that I'll say, oh, I should have said this. But for now, nothing really comes to mind that environmental factors can affect Alzheimer's onset. So the greatest risk is age. So basically, if you're over 65, that's basically the biggest risk for Alzheimer's.
Okay. For anybody who asked that question, there is Timothy's email. So if you want to send him a follow-up email, and in case there is some more information he can share. There is a question about SOLA mutation, SOLA mutations, and you'll have to explain what those are and if they're associated with early onset sporadic AD, Alzheimer's disease.
Right. I think you're asking that because you know the answer already. So SORLA is a risk gene. So basically, they're mutational variants that basically render individuals more prone to AD risk past age 65. And the answer is yes. So basically, there are early onset forms of SORLA, this gene, that have been identified in France that have basically made individuals more susceptible. So Chembel, I'll send you some references that of these early onset forms of SORLA so these mutations that can manifest in early onset.
So that's another angle that researchers could take to find out how to stop that mutation.
Right. And we're trying to find a difference between the mutations that give the late onset form and the early onset form. So, and it's not really 100% clear because it doesn't seem, although they've only tracked, families recently, so they don't know whether someone has an early onset form from each generation to generation, they can actually have 100% chance that this individual will have Alzheimer's. So as of now, I think once we have more information, we can say that these forms of early onset forms of SORLA are similar to these other genes such as APP that are 100% penetrant. So you will 100% have, these early onset Alzheimer's at age 40.
Thank you for that. And do you happen to know what the life expectancy for people with early onset Alzheimer's is?
That's a good question. I actually don't, that's a question I cannot answer. I'm not quite sure. However, basically the patients need to, their brain function needs to degrade to the point that certain faculties will disable their ability to survive. So this is something interesting that I found, that most Alzheimer's patients, they die from their inability to swallow. So basically they choke to death. So their mental capacity has degraded to the point that they cannot swallow properly. So basically that's how they most commonly pass away. So basically my answer to that is I don't know, but basically the brain function has to degrade to that level in order that survival drops to zero.
Thank you. And then are there people who have the variant APOE that you talked about earlier who do not develop Alzheimer's disease? Or is that a guarantee? If you show that variant, you will for sure?
Right, so they're doing that comparison experiment, and there's one variant, so that's the ApoE4 variant, that will increase your chances of AD onset. And this is by quite a bit. So I think one allele can increase your odds of AD onset by maybe 2 or 3-fold, or maybe even more. So there's actually a variant called E2 that is more protective. So if you have an E2 allele, then basically your chances of Alzheimer's onset drops. So the most of us, so we call the E3 allele, which is kind of in the middle, the most common allele. So most of us, we have this E3 allele. And so basically, there have also been other forms of apoE that have been identified, for example, a Christchurch mutation that is also protective. So even though an individual might have some type of Alzheimer's, so basically it's protective. I'll have to, think back on the paper that with the Christchurch mutation. But there are also other ApoE mutations that have been identified that are protective.
Oh, good. So there's the good and the bad, I guess.
Also APP. So there have also been APP variants that have been identified that are also protective as well. Yeah.
Thank you. With regards to what we can do, there's two questions that cover that. You had mentioned that inflammatory in the brain plays a role in Alzheimer's disease. What about anti-inflammatory foods? Should we be eating those? And if so, what are they?
Well, we should be eating those because they're good for us. So in terms of drugs, for example, that reduce, so there's been excitement over a potential that maybe anti-inflammatories can have some positive effect on AD. So they've actually tested non-steroidal anti-inflammatory drugs and their effects on modulating AD onset. They've had not so much success. So I imagine perhaps the same thing might happen with anti-inflammatory or antioxidants. I mean, we should just eat them because they're good for us. But in terms of whether that can promise reverse effects on Alzheimer's itself, that's kind of, a little bit uncertain. So, but in general, the better we take care of ourselves, there's probably a less chance that we'll have Alzheimer's disease.
Do you have one example of what qualifies as an anti-inflammatory food?
Anti-inflammatory food. So I hope I'm right in saying this, but oxidants will cause inflammation. So anything antioxidant might also be anti-inflammatory.
Great, thank you.
I hope I'm right in saying that.
I'm sure a little bit of research can help with that also. The other question on what we can do is sleep. We know that when we sleep, our brain does some cleanup work. So how important is sleep in Alzheimer's prevention?
Yes, that's absolutely important. So sleep, um, there have been recent publications that show that sleep enhances beta amyloid clearance. And so definitely, um, I think that proper sleep. So this is also been tested in animal models, so in mouse. So they show that basically the clearance of beta amyloid is not as efficient in mice that don't sleep properly. So definitely, the sleep cycle is important for proper clearance of beta amyloid.
That is good to know. Thank you. So at least 7 hours of sleep, everybody.
Yeah, that's right.
um, then we have a question about a new drug that is out on the market that has been in the news lately, and it's, oh, I'm going to mess that up. Aducanumab.
Yeah, I do cannon.
Thank you. And do you think it will be the game changer that some of the news articles suggest it will be?
Yeah, so I've been reading that too. So that's one monoclonal antibody I did not want to talk about. So I have an article here. If you want to send me an email, please do. But there's an article here that shows, I don't know if I can. So it says aducanumab, no change in rate of cognitive decline. So this is an article from Dennis Selko. So I think what we're seeing, on the internet and what the scientists think might be different. So I'm probably not the best person to answer this question, but I think the feeling is kind of mixed. I know that Biogen, their stock is going up because they're saying that there might be promise with this one beta amyloid targeting monoclonal, but um, according to some articles, maybe there's mixed reviews. Is
Mixed
maybe
reviews.
what I
Okay,
should say. Yeah.
thank you. You had mentioned earlier the mouse model memory recovery. Can you further clarify that?
So I'm not quite sure improving protein production in neurons. Trying to think if I can think of a specific article you're talking about. Um,
And maybe that's one where the participant can follow up with
yeah,
you.
that'd be good.
I will make sure that I send the email address around one more time. I think I
Yeah,
sent
please.
it twice, but I'll put it in the chat again. And maybe Dave, you can follow up with an email. That would be great.
Thanks.
So I'm going to move on to the next question. Is it true that loss of smell and taste is an indicator of Alzheimer's disease?
So I'm wondering if you're referring to the peanut butter test. So there was a article from a research group that suggests that perhaps one of the first affected regions in Alzheimer's patients is the olfactory bulb. So basically, where you have where you said smell. So basically, they say that Alzheimer's disease, you take a ruler, basically peanut butter at the end, and if you start to be unable to smell the peanut butter, then you might be diagnosed with Alzheimer's. So that's the last I've heard of that, and I'm not sure if that is actually a bona fide, finding, whether this is reproducible upon in all patients that. And I'm not sure if that would qualify as a diagnostic. So that's not saying it's not, but I've only heard that one story. So
So more research is needed
right,
on that to
we
make
need
sure
to
that it's
find out
prevented.
more. Right.
Yeah. Okay, so we have two more questions, which is great because then that brings us right up to our scheduled end of time of the event. Diagnostic testing for Alzheimer's disease, MRI or executive functioning?
So by the time that we start to have memory problems, there is a significant amount of A beta that's in our brains in a lot of these AD patients. So that is to say that this does not happen. So one thing I would like to bring up that really fascinated me when I joined the field is that there are actually individuals that have a ton of A beta in their brain, but they have zero cognitive decline. They're completely normal, and they can go for decades or the rest of their life with a lot of beta amyloid in their brain, but their cognitive function appears to be normal. So A beta itself as a diagnostic is not great. There are other markers that are closer, for example, tau. Once we have these modified forms of tau start to build up, that correlates more closely with cognitive decline. And of course, we're still on the race for biomarkers. So we're still trying to find a good biomarker that will indicate, if Alzheimer's is going to happen.
Thank you for that. And then one of the question is, if Alzheimer's disease doesn't show up until decades after the beta amyloids have started accumulating, would it be reasonable to start providing inhibitors for everyone so that they don't accumulate as much as they do?
Well, with inhibitors, especially the gamma secretase inhibitors that have failed in the clinical trials, is they target other pathways. For example, gamma secretase also cleaves Notch. So this is a very protein that's important in development. So right now, they're trying to find something called gamma secretase modulators. So something that will inhibit the beta amyloid formation, but it won't touch other pathways. And so if we can parse out, drugs that are specific to detrimental cleavage pathways, namely beta amyloid, then I think it's possible. Yeah, I mean, if everybody takes these drugs, then no more beta amyloid, then no more Alzheimer's. But you need to remember there are also other forms of dementia that are beta amyloid dependent. But I think it's a good idea because you're taking away that big proportion, 50 to 80% of are Alzheimer's disease. So that's a good.
Yeah, that would be amazing.
Good thought.
Timothy, thank you so much for joining us today. And for the audience, thank you so much for being here today, for engaging with us, for asking such wonderful questions and interesting questions. I hope we were able to provide you with some of the information you were hoping for. And I hope you found it enjoyable and as interesting as I did. With that, I want to say thank you, goodbye, and stay safe and stay healthy.